All right, now let's talk about Chapter 4. This is a survey of prokaryotic cells and microorganisms. We're going to largely be talking about bacteria in this chapter. We will touch on archaea at the end, and we're going to be talking about things like their structure, bacterial shapes and arrangements, and we'll even touch on some pathogens that we'll go over a little bit later in this course. Characteristics of cells and life. So all living things are made of cells that share some common characteristics. So cells are going to have a basic shape, either spherical, cubical, or cylindrical usually. Cells are going to have an internal content of cytoplasm surrounded by a cell membrane. And all living cells are going to have DNA in them, ribosomes, and metabolic capabilities, so the ability to use energy. There's two basic cell types we'll be talking about, eukaryotic and prokaryotic, and in this chapter we're going to be focusing on prokaryotic cells. So we've talked a little bit about the difference between eukaryotic cells and prokaryotic cells, but we'll kind of go over it again. So eukaryotes are going to be animals, plants, fungi, protists, basically any living thing other than bacteria and archaea. Eukaryotes contain membrane-bound organelles, and they also contain double membrane-bound nucleus. So they're very structurally complex cells. Prokaryotic cells, on the other hand, we said are going to be bacteria and archaea. They don't have a membrane-bound nucleus or any other membrane-bound organelles. They are much simpler cells. Okay, so all living things are going to be able to reproduce and show some sort of heredity, which is basically the ability to pass on one's genome to the next generation. These organisms are going to have a genome composed of DNA packed in either one or more chromosomes. We have multiple chromosomes. Many bacteria just have one. Organisms can produce offspring either sexually or asexually, depending on the organism. Living things are going to show growth and development. They're going to have metabolism. That's going to basically be the chemical and physical life processes that occur within the organism, essentially breaking down molecules into smaller molecules and building up molecules from small molecules, okay? How the organism uses its energy. Living things are going to be responsive. They show movement and or irritability. They can respond to stimuli within and without themselves. Many organisms are able to move. Living things have cell support, protection, and storage mechanisms, such as cell walls, vacuoles, granules, and inclusions, which we'll talk about here in just a little bit. Living things also are able to transport nutrients and waste. Okay, so this figure, there's a little bit to this figure that I want to explain. So it's showing essentially every possible structure that can be on a bacterial cell. So here you have what's found in all bacteria. So the cell membrane, the chromosome, the ribosomes, and the cytoplasm. Over here, all of these things are found only in some bacteria. And they're not all found in any one bacterial species. So this image is kind of the impossible bacterium. It has everything possible, which we know there's no species of bacteria like that. So I like to give that disclaimer there. What I want you to do with this figure is come back to it after we talk about these structures, and you can find where these structures are on this bacterium, and kind of read a recap of what each structure does. So we'll start talking about the structures here, and then we'll come back to this image at the end. Okay, so we're going to talk about the outermost structures first, and then work our way into the inside of the prokaryotic cell. So the first thing we'll talk about is appendages. And there are two major groups of appendages. There's appendages that help the organism move. That's going to be the flagellum and the periplasmic flagellum, plural flagella. This is also known as the axial filament. And then there's appendages used in attachment. So you've got fimbriae, which are going to help the organism attach to surfaces or to other cells. Then you have the pilus, plural pili, and this is used specifically for bacteria to attach to another bacterium and transfer genetic material, and that's a process called conjugation. We'll talk about that in more detail later on. Okay, so the flagellum, its primary function is motility. There are three distinct parts. You have the filament, which is like the tail-like portion of it, the part that you really think of when you think of a flagellum. And the filament inserts into the hook, which is essentially a sheath. It's a curved tubular structure that's attached to the cell by the basal body, and the filament fits into it. And then the basal body is a stack of rings that's anchored through the cell wall to the actual cell membrane. And it's this arrangement of the filament, the hook, and the basal body all together that allow the flagellum to rotate 360 degrees. Okay, so for example, here's the filament, right? The tail-like portion, and this would extend off this way. And you can see it inserts into the hook, which then connects to the basal body. And this arrangement is going to allow the filament to spin around 360 degrees, either in a clockwise or a counterclockwise direction. Now, the difference here between these cell envelopes we'll talk about here in a little bit, but this is a gram-negative cell envelope, and this is a gram-positive cell envelope. That's why these look a little bit different. Okay, so organisms can have either one flagellum or multiple flagella, and they can be arranged in a variety of ways. So you've got monotrichous. This is going to be a single flagellum at one end. You can see that here. Here's one flagellum coming off the end of this organism. You're going to have lophotrichous. This is small bunches emerging from the same site. So here you've got like a tuft of flagella coming out. Amphitrichous. This is going to be flagella at both ends of the cell. So you can see that down here in the corner. You have flagella coming out of this end and this end. And then you can have peritrichous. This is flagella dispersed over the entire cell surface. And you can see that in this example here. Now, I also have another slide. This again shows the different flagellar arrangements. And I would be familiar with these because there's likely to be a test question over them. Okay, so how does the organism actually move? Well, the flagella are going to guide the bacteria in a direction in response to an external stimulus. So if it's in response to a chemical stimulus, this is called chemotaxis, and there's positive and negative chemotaxis, and I'll explain the difference here in a second. If it's in response to a light stimulus, this is called phototaxis. And what happens is the stimulus is going to cause the flagella to move either clockwise or counterclockwise. And which direction it moves is going to result in either a run, which is a smooth linear direction if it's counterclockwise, or a tumble if it's moving in a clockwise direction, where basically the organism stops moving in the direction it was moving in and changes direction. Okay, so over here you can see a tumble, a run, and a tumble. And this is how it's represented on the diagram over here. A tumble is a red dot, a run is a green line, and then another red dot here for this tumble. And what happens is the flagella are going to turn in one direction, causing a run. And then when the flagella reverses direction, it's going to result in a tumble. So here you have a run, you have a tumble, you have a run, a tumble, run, tumble. And you can notice there's frequent tumbles. There seems to be no direction to this movement. It's just kind of random. And this is typical when there's no attractant or repellent in the area. Now compare that to when there is an attractant in the area. Now this diagram is showing a gradient of attractant, meaning that there's more attractant on this side and less attractant on this side. And what you're going to see is fewer tumbles and longer runs. And this ultimately is going to result in movement toward the stimulus here, because this is an attractant. Now, if you have movement toward a stimulus, that's called positive chemotaxis. If the bacterium or organism is moving away from the stimulus, that's going to be negative chemotaxis. All right, periplasmic flagellum. You'll find these in spirochetes, which are corkscrew-shaped bacteria. And spirochetes possess this wriggly mode of locomotion that is caused by these periplasmic flagella. And all these really are is just internal flagella, okay? They're also called axiofilaments. And here, an internal flagellum is enclosed in the periplasmic space, I'll show you that here in just a second, between the outer membrane and the cytoplasmic membrane. And this twisting, flexing, coiling motion that the periplasmic flagella impart is what aids the bacteria in penetrating tissues. All right, so here we have a spirochete. And you can see there's these internal flagella running along the length of the spirochete. And where exactly these flagella are located, you have to zoom in a little bit. And you can see here is the cell membrane. This is the cell wall. And then here's the outer membrane. And we'll go into detail with these, what these different membranes and what the cell wall means here shortly. But I just want you to know that inside of this organism, there is this space, and this is called the periplasmic space. And that's where this internal flagellum is located, okay? So big picture stuff, this is an internal flagellum or internal flagella. And it causes the spirochete to twist and wriggle in such a way that it helps it penetrate tissues. And we'll learn about some spirochetes in this course, one in particular that causes syphilis, and how its ability to invade tissues really makes it a significant pathogen. Okay, a couple of questions here. What is the primary function of bacterial flagella? Well, recall the bacterial flagellum is involved in movement, so the answer here is going to be C, to provide motility. What is the role of the basal body in the structure of a bacterial flagellum? Well, if we go back and look, you can see here that the basal body is the anchor to the entire apparatus. So the answer is B. How does the movement of bacterial flagella contribute to the motility of bacteria? Well, remember that flagella are going to rotate in either a clockwise or a counterclockwise direction in order to propel the bacterium. So the answer here is going to be B. What is the difference between a bacterial flagellum and the periplasmic flagella found in spirochetes. Well, remember that the periplasmic flagella are going to be inside that periplasmic space, and they kind of wrap around the organism here. So the answer there is A. Fimbriae. These are fine hair-like bristles that emerge from the cell surface. You can see them here. They're going to aid in adhesion to other cells and to surfaces, okay? So when you think fimbriae, you need to be thinking adhesion. These are basically very sticky little hair-like projections, okay, that help the organism stick to other things. Okay, pili, or the singular is pilus. A pilus is a flexible tubular structure. It's made of pilin protein. This is found only in gram-negative cells, and what it does is it functions to join bacterial cells, allowing them to transfer, at least partially transfer DNA. And this process is called conjugation. So you can see here, here's one cell, here's another cell, and you can see a pilus coming out and extending between the cells. Here, this sort of hazy, dark area halo around the cell is actually tons of tiny little hair-like bristles, which we call fimbriae. Okay, so we covered bacterial appendages. Now let's kind of talk about the bacterial cell itself from outside to inside. So from on the outermost layer is going to be this thing called the glycocalyx. And all that is a coating of molecules on the farthest outermost portion of the cell, outside of the cell wall. And these molecules are going to consist mainly of sugars and proteins. Now, the glycocalyx can take on one of two types. It can be a loose slime layer, which is loosely organized and loosely attached, or it can be a highly organized, tightly attached capsule. And what these things do is they help the cell prevent losses, prevent nutrient loss, prevent loss of water. They're also going to help protect the cell as well. So here we have the two forms of glycocalyx. You can have this loosely organized slime layer on the outside of the bacterial cell, or you can have sort of this tightly organized capsule on the outside of the cell. Functions of the glycocalyx. So I mentioned this is going to protect these cells from dehydration and loss of nutrients. Another thing that the glycocalyx is going to do, specifically the capsule, is it's going to inhibit phagocytosis by white blood cells, contributing to pathogenicity. Okay, what that means. Phagocytosis is something that our white blood cells do to pathogens. They essentially open up and eat the pathogen, taking that pathogen into themselves and then destroy that pathogen. The capsule prevents these white blood cells from being able to eat them, okay? From being able to phagocytose them. And that's going to contribute to the bacterium's pathogenicity, meaning its ability to cause disease. Okay, and the glycocalyx is also going to function in attachment. And this is important in the formation of biofilms. Now, what exactly is a biofilm? Well, a biofilm is essentially a micro ecosystem of different bacterial species that are all living kind of in the same community, okay? And it starts when bacteria attach to a surface and divide and multiply and form a colony on that surface. And as they do, they secrete materials that form sort of this gelatinous matrix that the different species of bacteria can live in. Okay, and that is a biofilm. Biofilms are troublesome because they are difficult to treat. They like to form on medical devices such as catheters, and they are difficult to penetrate with antibiotics. So treating with things like IV antibiotics usually is of no use. They're also difficult to remove physically. So you just have to take out the medical device and replace it. So biofilms are a big problem in the healthcare world. Okay, this is just kind of showing you different bacterial colonies, not particularly zoomed in or anything. And you can see these white streaks here are going to be bacterial colonies, bacteria who don't have capsules, whereas these big globs, these kind of mucoid appearing colonies, are going to be made up of bacteria that do have capsules. The cell envelope is the covering outside of the cytoplasm. Remember, the cytoplasm is that liquid substance inside of the cell. The cell envelope is basically composed of two layers, okay? The cell wall, which is outside, and the cell membrane, which is more internal to the cell wall. What the cell envelope does is it maintains cell integrity. Basically, it keeps it from breaking apart. And you'll learn there are two different groups of bacteria that we're going to talk about, gram-positive and gram-negative bacteria, and they're demonstrated by the gram stain, which we'll be doing in this course as well. They have different cell envelopes. The gram-positive bacteria have a very thick cell wall, and then they have a cell membrane. The gram-negative bacteria, they have a thin cell wall, and as well as a cell membrane, but they also have an extra outer membrane. And I'll show you what I mean here. Okay, on the left here, we have a gram-positive bacterium. On the right here, a gram-negative bacterium. And we're looking at the cell envelope. Here, you're looking at the cell membrane, also called the cytoplasmic membrane, also called the plasma membrane. Moving further outside of that, you're going to have this thick cell wall. Remember, gram-positive is going to have a very thick wall. Go over here to gram-negative, you can see it also has a cell membrane. It also has a cell wall, but notice how thin it is. And then notice it has an additional membrane outside of all of that. Now this additional membrane has molecules in it. And the one that I want to really talk about is called lipopolysaccharide. We also call this endotoxin, and you'll learn that when lipopolysaccharide is exposed to mammals, mammals develop a very specific immune response. And we are no exception. So what is the cell wall in bacteria made out of? It's made out of this compound called peptidoglycan. And what this is it's a repeating framework of long sugar chains that are cross-linked by short protein fragments. And you can see right here, here's a sugar chain. Here's another sugar chain. And then here is this green line is a protein fragment that is connecting these sugar chains. And you put enough of these together and it provides a strong and flexible framework. And peptidoglycan is what gives the cell wall its rigidity. Why is this important to know? Well, many antibiotics target the cell wall. So if you can break down the cell wall, you can potentially kill the pathogen. Okay, the gram-positive cell wall. Remember, we said the gram-positives have a thicker peptidoglycan cell wall layer than gram-negatives. And you can see here 20 to 80 nanometers thick, whereas gram-negative organisms typically have peptidoglycan cell walls that are 5 to 10 nanometers thick. The gram-positive cell wall has teichoic acid and lipoteichoic acid in it. These molecules function in cell wall maintenance, enlargement during cell division, and they stimulate the innate immune system. Some cells are going to have a periplasmic space, and that's going to be between the cell membrane and the cell wall. So here you can see the gram-positive organism. We have the plasma membrane or the cell membrane, the periplasmic space between the cell membrane and the cell wall here, which is composed of peptidoglycan. You can see teichoic acid and lipoteichoic acid are embedded in the peptidoglycan. The gram-negative organism is going to have an inner and outer membrane, and the space between that is called the periplasmic space, and it's going to contain a thin cell wall, a thin peptidoglycan layer. I already mentioned that the outer membrane contains lipopolysaccharides. The lipid portion of lipopolysaccharides is called endotoxin, and it can become toxic when released during infections. It can cause septic shock. Lipopolysaccharide can also function as receptors in blocking immune responses. Lipopolysaccharide can suppress the adaptive immune system. It can also disrupt cell-to-cell interactions between immune cells, and so it can cause some blunting of the immune response. The outer membrane also contains porin proteins, and porin proteins are going to regulate molecules coming and going from the bacterium. For example, antibiotics. So porin proteins can regulate the permeability of antibiotics. Porins can affect a bacterium's antibiotic resistance by either allowing or blocking the passage of antibiotics into the cell. Here we have the gram-negative organism. You can see the cell membrane here, the outer membrane here, and the space in between is the periplasmic space, which contains a thin cell wall composed of peptidoglycan. You can see the porin molecules here in the outer membrane, and it also contains lipopolysaccharide. Okay, which of the following statements about the cell wall of gram-positive bacteria is true? A, it contains a thin layer of peptidoglycan and an outer membrane. Nope, that's gram-negative. B, it is composed mostly of lipids. No. C, it contains a thick layer of peptidoglycan and teichoic acids. Or D, it lacks a cell wall entirely. Well, remember, gram-positive bacteria do have a cell wall, and it's going to be a thick layer of peptidoglycan, not lipids. And they also, the cell wall also has teichoic and lipoteichoic acid in it. So C is the answer here. Which of the following is found only in gram-negative bacteria? A, peptidoglycan. No, that's found in both gram-positive and gram-negative cell walls. B, teichoic acids. Remember, those are going to be found in gram-positive. We're looking at gram-negative here. D, the cytoplasmic membrane. Remember, this is also the cell membrane. And both gram-negative and gram-positive organisms are going to have a cell membrane. So the answer here is C, lipopolysaccharide. This is also called endotoxin. So what exactly does it mean to be gram-positive or gram-negative? We've talked about the literal structural differences between gram-positive and gram-negative organisms. But what does gram even mean? Well, Hans Christian Gram was this bacteriologist in the 1800s who developed a staining technique. And this staining technique would stain gram-positive organisms purple and gram-negative organisms red. Why do we need to stain microorganisms? Well, if you just looked at them as they are under the microscope, they're so thin and so see-through that you're not going to be able to distinguish any of their structures. So we add dyes to them so that we can see better under a light microscope. The gram stain is actually a fairly straightforward procedure. What you do is you start by staining all the cells with crystal violet, and that's going to turn all the cells purple. Then you're going to add iodine. Iodine is the mordant, and what that means is it helps that purple dye stick deep within that thick peptidoglycan layer of gram-positive organisms. Next, you're going to add alcohol. Alcohol is going to break down the outer membrane of the gram-negative organisms, and remember they have a very thin peptidoglycan cell wall that really doesn't hold on to the crystal violet very well. So alcohol is going to decolorize the gram-negative organisms. It's going to make them look clear again. The gram-positive organisms are not going to be affected in that way. They're still going to look purple. Finally, because we need to be able to see the gram-negative organisms too, we add a second dye. It's called a counterstain, and it's safranin. And what that does is that stains the gram-negative organisms red or pink. The gram stain is central to bacterial classification and identification. One of the very first things you want to know about a bacterium, a bacterial infection, is whether it's caused by a gram-positive or a gram-negative organism, because that's going to shape your treatment. It's going to shape which antibiotics you choose and which structures of the cell envelope you're going to be targeting. Okay, so here we go. Step 1, crystal violet. This is the primary stain. It's added to the sample and it stains all the cells, the gram-positive and the gram-negatives, purple. Next, you add iodine. This is the mordant, and it's going to make the dye less soluble, so it's going to adhere to those cell walls. It's going to get really stuck in those thick gram-positive cell walls during this step. Step 3, you add alcohol. This is the decolorizer. It's going to wash the primary stain away from the gram-negative cells, only the gram-negative cells. Now you can see they don't have any stain whatsoever. The gram-positives stay purple. Now we use the counter stain and stain all the gram-negatives red. Some bacteria are going to have atypical cell walls. For example, Mycobacterium and Nocardia. Now these guys have a gram-positive cell wall structure, but they have these lipid mycolic acids coming off of the cell wall. These mycolic acids make these species really resistant to our immune system and our immune defenses. It also makes them resistant to most chemicals and dyes. So we use a totally different stain for these organisms. We use the acid-fast stain, okay? So just know that in addition to the gram stain, there are other stains out there that we use to diagnose species. Some bacteria have no cell wall, for example, Mycoplasma. Their cell membrane is going to be stabilized by sterols instead. And because they don't have a cell wall, they exhibit pleomorphism, which is a variation in cell shape and size that you see within a single species. And some species are noted for their pleomorphism, such as Mycoplasma. Okay, so we're still on the cell envelope. Remember, we talked about the outer membrane in gram-negative species and how it has porins and lipopolysaccharides. We talked about the cell wall in gram-positive and gram-negative bacteria, being thicker in the gram-positive and thinner in the gram-negatives. And now we're going to talk about the cell membrane, which both gram-positive and gram-negative organisms have. The cell membrane is going to be composed of a phospholipid bilayer, and I'll show you what that looks like in a second, with proteins embedded in it. And the way that these proteins move throughout the phospholipid bilayer is called the fluid mosaic model, and I'll talk a little bit more about that in a minute. The cell membrane is going to provide a site for energy reactions. Remember that the bacteria don't have membrane-bound organelles to process energy reactions, so they're going to use their cell membrane. They are also going to use their cell membrane to process nutrients and for synthesis of proteins and things like that. The cell membrane also functions in passage of nutrients into the cell and discharge of wastes from the cell. And it's selectively permeable, meaning it lets some molecules in, but not others. Okay, here's that phospholipid bilayer. And what a phospholipid is it's this molecule here that's composed of this head and these two tails coming off of it. Okay, and the head is hydrophilic. That means it likes to interact with water. While the tails are hydrophobic, it prefers not to interact with water. So these molecules naturally align themselves to where the heads are pointing out towards the intracellular fluid and the extracellular fluid, and the tails are pointed inward toward each other. This creates a natural barrier to the external environment. It's not just phospholipids that make up the cell membrane, though. The cell membrane has different proteins and different receptors, things like that, embedded in it. And they kind of float in this phospholipid matrix. And we call that the fluid mosaic model. So while you have a protein here, just know it's not necessarily anchored in place. It's kind of free moving throughout the phospholipid bilayer. Okay, now we're going to move inside the bacterial cell into structures and substances that are create the bacterial cell's internal environment. So first we have this cytoplasm. This is going to be that gelatinous solution of sugars, amino acids, and salts that make up the inside substance of the cell. It's 70 to 80% water, okay? And that's going to serve as a solvent for the materials that are used in all of the cell's functions. Next, you have the nucleoid, and this is going to be the region inside the cell that contains the DNA of the chromosome. Now, bacteria typically have a single circular chromosome. It's double-stranded DNA. But there are some exceptions to that. There's some bacteria that have more than one chromosome. Some bacteria that have linear chromosomes. So, but by and large, it's going to be a single circular chromosome. It's a double-stranded DNA molecule that contains all the genetic information of the organism. Now, you've got, in addition to the chromosome, these things called plasmids. These are free, small, circular, double-stranded DNA molecules that are not essential to bacterial growth and metabolism. Instead, plasmids tend to convey resistance genes, and bacteria like to share plasmids between one another. We like to use plasmids in genetic engineering because we can manipulate them. We can tell them to express a certain gene, for example, the insulin gene, and we can transfer plasmids from cell to cell. Okay, looking at this image, you can see several of the things we've already talked about. For example, here's a flagellum coming off of the bacterial cell. Here is a pilus. You can see the fimbriae, the hair-like projections from the bacterial cell surface. You can see the glycocalyx here in the form of a capsule. Then you've got your cell wall. And then innermost, you have the cell membrane. And then you have the cytoplasm and all the stuff inside of the cytoplasm. In particular, here's the nucleoid. Now, this DNA is kind of all coiled up on itself, but if you were to take it and stretch it all the way out, it would be one very large circular strand of DNA. Down here you have a small circular plasmid, and remember the plasmids are going to contain primarily resistance genes. And then here is an inclusion body, and we'll talk about that here in just a second. Another thing to notice here is these small dots throughout the cytoplasm. These are ribosomes. Okay, and ribosomes are going to be the site of protein synthesis in the cell. The bacterial ribosome is made up of ribosomal RNA and protein, and it's going to consist of two subunits, a large and a small subunit. You'll see a very similar scheme in the eukaryotic ribosomes. They also have two subunits, large and small, but they are slightly different size. Ribosomes, like I said, are where proteins are made, and they're found in all living cells. Okay, so here is a diagram showing the small subunit and the large subunit of the ribosome. You put them together, you have your ribosome. These numbers indicate the type of subunit. So in bacteria, the small subunit is designated 30S, the large subunit is designated 50S, and the ribosome as a whole is designated 70S. The S stands for sedimentation, and what they do is they basically take the small subunit and they let it sink in a glass of liquid, and they note how long it took for it to settle. They do the same thing with the large subunit, and then they do the same thing with the ribosome. And these numbers are not additive necessarily, because when you put the small subunit and the large subunit together, the sedimentation is going to depend on the density of this molecule, on the shape of this molecule, etc. So that's why these numbers don't add up. What you need to remember is that the bacterial ribosome is 70S, and you need to know the designation of each subunit. So the small subunit is 30S, and the large subunit is 50S. Next, we have inclusions and granules. These are intracellular storage bodies. They're going to vary in size, in number, and in what they contain. And the bacterial cell is basically going to use them when its environmental sources are depleted. So again, these are just terms for storage bodies. Okay, the cytoskeleton. This is going to be an internal network of basically protein strands that are connected to the cell membrane. And they're going to give structure and shape to the cell, and they're also going to help facilitate cell division. Okay, so endospores. We've touched a little bit on endospores. Let's talk more about them. These are going to be inert. They're resting cells that are produced by some gram-positive genera. Primarily, we'll talk about Clostridium and Bacillus. Now, these species have a two-phase life cycle. They can be a vegetative cell, where they're metabolically active and growing. This is what we think of when we think of a bacterial cell. Or they can be in an endospore, which is an inactive version of the organism. Endospores occur when the organism is exposed to an unfavorable environment, okay? So things get really hard. The organism sees that things are becoming more and more difficult, so it produces endospores that can survive these difficult conditions. Endospores are capable of high resistance and very, very long-term survival, millions of years. Now, when the environment becomes favorable again, the endospore is going to become a vegetative cell again. Okay, so we have sporulation and we have germination. So sporulation is going to be when the bacterium decides to form endospores. Endospores are the hardiest of all life forms. They're dehydrated, they are metabolically inactive, and they can withstand extremes in heat, drying, freezing, radiation, and chemicals, okay? So these guys are really difficult to kill. We mentioned their longevity, millions of years. Now, these are not spores that are produced for reproduction. These are spores that are produced for survival, okay? How do we kill them? We use pressurized steam at 120 degrees Celsius for 20 to 30 minutes. Next is germination, and this is when the endospore returns to vegetative growth when the environmental conditions become favorable again. Okay, so here's that image again, and here it's listing all the structures that we've talked about. I encourage you to go through here and make sure that you are familiar with all of these. Really know, when it comes to the cell envelope, know the layers in order. So you need to know cell membrane and then cell wall, and then if it's a gram-negative organism, outer membrane, okay, in that order. For the items on here that we have not talked about, don't worry about that. I'm not going to test you over that. I'm only going to test you over the things that we've discussed in lecture. Okay, if you discover that the bacterium you're studying stores phosphate, which of the following would you expect to contain the stored phosphate? Well, let's just start from the bottom and work our way up. So endospores, no, remember these don't store anything. They are forms of an organism that are used for survival, okay? Lipopolysaccharides, no, that's a molecule found in the outer membrane of gram-negative organisms. Plasmids, remember these aren't storage bodies either. They are circular pieces of DNA. Ribosomes are not storage bodies either. They are the site where proteins are made. So that must mean that the answer is A, granules. And if we go back, you can see the two storage bodies we're talking about are going to be inclusions and granules. Okay, on to bacterial shapes and arrangements. So bacteria are going to vary in shape, size, and arrangement, but typically we can describe them by one of three basic shapes. So we can, if they're spherical, describe them as a coccus. If they are rod-like, so longer than they are wide, they're going to be a bacillus. And if they're helical or twisted, they're going to be a spirillum. And then underneath bacillus, we have the coccobacillus. This is going to be a short and plump rod. And we have a vibrio. This is a gently curved rod. Underneath the spirillum, the most common helical or twisted microorganism we're going to talk about is going to be a spirochete. Okay, so if the cell is spherical, the bacterium is going to be described as a coccus. The plural is cocci. But there's some variation in how spherical these cells are. So they can be oval, kind of bean-shaped, or even pointed, okay? This photo here is of Staphylococcus. And I like to go along, I like to learn what the names mean as I go along. So staph is going to be from the Greek staphyl, meaning cluster of grapes. And of course, coccus means berry. So this is so named because it resembles clusters of grapes or bunches of grapes under the microscope. Next, you have the rod or the bacillus. So this is going to be a cell that is cylindrical. I want you to note there's also a genus named Bacillus, so don't get these confused. Lowercase b, Bacillus, is talking about the shape. Capital B, italicized Bacillus, is talking about the genus. Rods are going to be varied in their actual form. Depending on the species, they're going to be blocky, spindle-shaped, round-ended, drumstick-like, long and thread-like, club-shaped, etc. When the rod is short and plump, we call it a coccobacillus. And this photo here is of Legionella pneumophila. Pneumophila from the Greek lung-loving. So as you can imagine, Legionella causes lung diseases, lung infections. Vibrio, these are gently curved rods. What you see here is Vibrio vulnificus, the Latin causing wounds. So Vibrio vulnificus, among other things, can cause skin infection. A bacterium with a slightly curled or spiral-shaped body is going to be a spirillum. This is from the Latin, meaning little coil. Spirilla have a rigid helix. They're twisted twice or more along its axis, like a corkscrew. This is in contrast to a spirochete, which is another spiral cell. But this one is more flexible than the spirillum, okay? When it comes to the test, you are not going to be tested on spirilla. You will be tested on spirochetes because we're going to talk about spirochetes in this class, okay? Next are branching filaments. So a few bacteria are going to produce multiple branches off of the basic rod structure. What you can see here is the basic rod structure, and here's a branch coming off of it, then another branch, etc. This is called branching filaments. You see this in species like Streptomyces. Strep meaning twisted or chain, and myces meaning fungus. The reason that this organism got its name is because of this long filamentous branching pattern. It resembles the hyphae of fungi. So they thought initially that Streptomyces was a fungus. So that was shapes. Let's talk about how bacteria arrange themselves. So for the cocci, right, the spherical bacteria, they can be arranged by themselves in pairs. This is diplococci. In groups of 4, that's tetrads. In irregular clusters, such as Staphylococcus. In chains, such as Streptococcus. Remember, strep means chain or twisted. And in cubical packets called a sarcina. Bacilli have fewer arrangements that they can take on. They can be a pair, so diplobacilli. They can be in chains, streptobacilli, and then they can be in this arrangement called the palisades, and I'll show you that here in a minute. So how do they get in these arrangements? Well, it depends on what plane they are dividing in. Division in just one plane is going to result in the daughter cell basically being lined up next to the parent cell. If you divide over and over and over again, that's going to cause a chain. Okay, so this is how we get Diplococcus and Streptococcus. Now, if the cell divides in 2 perpendicular planes, that's how you're going to get a tetrad or a sarcina, okay? And a sarcina is essentially just a cubic arrangement. Division in several planes is going to result in irregular clusters, okay? So that's where you're going to see Staphylococcus. Arrangement of bacilli, we said, could be single, they could be in pairs, they can be in a chain, or this arrangement called palisades. This is where cells of a chain are going to remain partially attached to one another by a small hinge region at the ends. And this is what the palisades looks like here. You can see hinge region where it's still attached. Spirilla and spirochetes, for all intents and purposes, just think of these as rarely remaining attached after cell division, okay? At least the ones we're going to talk about, they tend to be found in singles. All right, this image is trying to give you an idea of the relative size of bacteria. So here we're looking at a human hair, and it's been magnified 200 times, okay? Next to that, you see this clump of things. So let's zoom in on that clump of things. Now we've zoomed in 2,000 times. We can see a pollen particle, a white blood cell, a red blood cell, a yeast cell, and then we can see some smaller things here, not entirely sure what they are, so let's zoom in some more. Now we've zoomed in 20,000 times. Here you can see this rod-shaped bacterium is E. coli. You can see these spherical bacteria are Staphylococcus. And then even smaller next to them, you can tell, are the viruses. Okay, moving on to classification systems for prokaryotes. So here are the kind of the characteristics that we're looking at when we go to classify these microorganisms. So one, the microscopic morphology, which we just talked about a little bit. What kind of shape are the cells? What's the arrangement? Macroscopic morphology, this would be colony appearance. And you'll learn in the lab, colonies can look very different from one another. They can have different colors. They can have, different levels of shininess. They can be different sizes. 3, bacterial physiology. For example, does the bacterium produce its energy using oxygen? That would make it aerobic. Or does it produce its energy without oxygen? That makes it anaerobic. We can also do serological analysis. This would be detecting bacterial antigens. And of course, we do genetic and molecular analysis as well. All of these help us determine where exactly to place each species of bacterium in relation to one another. Okay, so there's this thing called Bergey's Manual of Systematic Bacteriology. It's a 5-volume resource that covers all known prokaryotes. So that means it's going to involve just 2 of the 3 domains, right? Prokaryotes are archaea and bacteria. And it's got 5 major subgroups with 25 different phyla. Each phylum is based on something different. So for example, shape, metabolism, genetic similarities, etc. So organisms with similar characteristics are going to be placed in the same phylum. The very first thing that we're going to do when we look at Bergey's manual is we're going to say, is this a gram-positive or a gram-negative organism? So right there, that's the first division. From there, the organism is going to be sorted into different phyla depending on its characteristics. And then it's going to be further classified into class, order, family, and finally genus and species based on those characteristics. Okay, and then just to kind of give an example here in the domain bacteria, how they split them up into different phyla. You can see here this phylum is for gram-negative cell walls, whereas these 2 phyla are categorized based on what the guanine and cytosine content in their genome is. Okay, this is another example of a phylogenetic tree, just showing the different relationships between the different organisms. You have the bacteria here, and then on this side you have archaea, and then eukarya. Okay, so some of the medically important bacteria that we're going to talk about in this course include the bacteria on this list, and I'm just going to kind of go over them quickly. So first we'll talk about gram-positive bacteria, and we're going to learn about the cocci in clusters, primarily Staphylococcus, okay? Staphylococcus known for its skin infections that it causes. Cocci in pairs and chains that are facultative. So let's go back here we're talking about bacteria that are aerobic or facultative. Aerobic means that they require oxygen to produce energy. Facultative means that they can produce energy in the absence of oxygen as well as in the presence of oxygen. Okay, so cocci in pairs and chains that are facultative, we think of Streptococcus, and this is the species that causes strep throat, dental cavities, and some other sequelae that we'll talk about in more detail when we get to it. Next, we have anaerobic cocci. So these guys have to produce their energy in the absence of oxygen. We'll talk a little bit about endospore formers. So bacillus, where you get anthrax from, Clostridium, which causes tetanus, gas gangrene, and botulism, and then Clostridioides, its main disease is C. diff. We'll talk a little bit about non-spore forming rods, such as Lactobacillus. This is actually one of the helpful bacteria in our body. We'll talk about Listeria, which can cause food poisoning. You have bacteria involved in acne, the cause of diphtheria, and then Mycobacterium, the cause of tuberculosis, and Hansen's disease, which is also called leprosy. More in the non-spore forming rods, we have Nocardia. This can cause lung abscesses, Actinomyces, which can cause dental infections, and Streptomyces, which is actually an important source of antibiotics. Okay, and then we're going to talk about gram-negative medically important bacteria. For example, Neisseria, species in this genus, can cause gonorrhea and bacterial meningitis. We have Pseudomonas. We'll actually work with Pseudomonas in the lab. This can cause a pneumonia and can also cause infections, especially in burn patients. We have other aerobic rods such as Brucella, Bordetella, Francisella, Coxiella, and Legionella. Facultative rods and vibrios. So we have Escherichia, right, of E. coli. We have Salmonella, Shigella. Both of those cause gastrointestinal disease. Klebsiella we'll work with in the lab. Proteus we'll work with in the lab. And then there's Yersinia, Yersinia pestis being the species that causes plague or the Black Death. Next you have Vibrio. Species in this genus are going to cause cholera and food infections. Talk about Campylobacter, which causes gastroenteritis, and Helicobacter, which causes gastric ulcers. Can also cause stomach cancer if left untreated. Treponema, aka syphilis, we're definitely going to talk about. And Borrelia, which causes Lyme disease, as well as Leptospira, which can cause a kidney infection. There are also bacteria that can become intracellular parasites. Some of them have to be inside of a cell in order to live. Some of them are facultative, so they can take it or leave it. Examples here would be Rickettsia, which is the cause of Rocky Mountain spotted fever, Ehrlichia, Chlamydia, and Bartonella. Bartonella causes cat scratch fever. And then finally we have bacteria with no cell walls. So we talked about gram-positive, gram-negative, and now this is an example of a bacteria with no cell wall, Mycoplasma, which can cause pneumonia, specifically a walking pneumonia. We'll talk about these pathogens in more detail when we get to the chapters that go over the diseases that are caused by these guys. Okay, what do we mean when we say species and subspecies? So a species, at least when it comes to bacteria, is going to be a collection of bacterial cells which share an overall similar pattern of traits in contrast to other bacteria whose patterns are going to differ significantly, okay? And the reason we use this definition is because bacteria are multiplying quickly and they are accumulating mutations. So it's not uncommon for bacteria of the same species to look a little bit different depending on the mutations that they've acquired. When they look different enough, that's when we call it a strain or a variety. This is a culture that's derived from a single parent. So this is a group of bacterial cells, which was derived from one single bacterial cell. And it's going to differ in its structure or metabolism from other bacterial cells of the same species. You can also have a different type. This is going to be a subspecies that can show differences in either antigenic makeup, susceptibility to bacterial viruses, or in its pathogenicity or its ability to infect. For all intents and purposes in this class, we're going to say subspecies, strain, variety, type. All of those are kind of interchangeable. We're not going to focus too much on the small differences between those terms, okay? Okay, question. Round bacterial cells growing in irregular clusters would best be described as, well, they're round, so you know it's going to be a coccus. And an irregular cluster is a staphule, so we're looking for staphylococci. What term describes a chain-like arrangement of spherical bacteria? Spherical again, so we're working with cocci. Chain-like arrangement, so chain is going to be strep, okay? So streptococcus is the answer here. Okay, prokaryotes with unusual characteristics. So these examples here are free-living, non-pathogenic bacteria. So they kind of mind their own business. We have photosynthetic bacteria. These guys can use photosynthesis and can synthesize the required nutrients from inorganic compounds. For example, cyanobacteria, also called blue-green algae. These guys have gram-negative cell walls. And they have chloroplasts with a lot of thylakoids in them, essentially the site where photosynthesis occurs. So they are heavily equipped for photosynthesis. Next, you have green and purple sulfur bacteria. These guys are also photosynthetic, but they don't give off oxygen as a product of photosynthesis. Instead, they give off sulfur. There are gliding bacteria, which are able to move across surfaces without the use of a flagellum. And there are fruiting bacteria. Fruiting bacteria are bacteria with a very complex life cycle, which involves the formation of aggregates of bacterial cells that differentiate to form reproductive structures. So, you know, the overall theme here is that prokaryotes are extremely variable. Obligate intracellular parasites, I already mentioned, the rickettsias. These are very tiny gram-negative bacteria that are obligate pathogens, intracellular pathogens, so they cannot survive outside of the host cell. Rickettsia rickettsii is what causes Rocky Mountain spotted fever, and this is transmitted by ticks. We'll learn about this more later. Here is an example of a cell that has been infected with rickettsial cells. You can see the smaller kind of ovoid shapes here being the cells of rickettsia. Chlamydia is also an obligate intracellular parasite. Chlamydia is also tiny. It is not transmitted by arthropods like the ricketsias. It is transmitted by close contact. Chlamydia trachomatis is the cause of a severe eye infection, and it's the cause of one of the most common sexually transmitted diseases. Chlamydia pneumoniae causes lung infections. Finally, just a little bit about domain archaea. These are the other prokaryotes, right? So we have bacteria, and then we have archaea. Archaea are actually more closely related to us than to bacteria, more closely related to eukaryotes than to bacteria. They, unlike bacteria, do not have peptidoglycan in their cell walls. And this is just a chart showing some of the different characteristics among bacteria, archaea, and eukarya. So the cell type, bacteria and archaea, are both going to be prokaryotes. Eukarya is eukaryote, okay? Chromosomes, bacteria can have a single or a few, and they're generally circular. Archaea have single chromosomes, one single chromosome, and it's circular. Eukaryotes have several chromosomes, and they are linear. Ribosomes, remember that bacteria has a 70S ribosome. Eukaryotes have an 80S ribosome. We'll learn about that I believe, in the next lecture. Archaea have a 70S ribosome, but the structure is more similar to that of the eukaryotes. We already talked about this, presence of peptidoglycan in the cell wall. Only bacteria have that, okay? So what do the archaea do? What makes them so different and so special? Well, they are able to live in the most extreme habitats in nature. And for this reason, we call them extremophiles. They are adapted to extreme temperature, very high salt concentrations, very acidic environments, and they're also adapted to pressure. These extremophiles are going to include methane producers, hyperthermophiles, so heat lovers, extreme halophiles, or salt lovers, as well as sulfur reducers. Really, this is probably going to be the last that we talk about archaea much in this course. Just know that they tend to be able to live in very extreme environments. Okay, organisms in the domain archaea have peptidoglycan in their cell wall. True or false? Well, remember, this is archaea, and only bacteria have peptidoglycan, so this one is false.